Hello everybody, I am Chala with Dr. Gilman Su from the Hong Kong Polytechnic University. The topic of my presentation today is determination of the functional impact of overexpressed PHOP gene on mycobacterial virulence using Zebrafish model. This is the content of my presentation. It includes introduction, aim of the study, methodology, results, conclusion, and acknowledgement. If I start with introduction with tuberculosis, tuberculosis is an infectious disease caused mainly by Mycobacterium tuberculosis, and the disease mainly affects the lungs, where it causes pulmonary tuberculosis, and it also affects the other body parts, including the central nervous system, where it causes the severe form of tuberculosis, which is called tuberculous meningitis. And even though the different control measures have been taken, the disease remains one of the most devastating public health problems globally. And the severity of this disease is mainly due to the increased virulence of Mycobacterium tuberculosis, which is the causative agent of the disease. And recently, we identified genetic mutations specific to those hypervirulent Mycobacterium tuberculosis, which is not found in low virulent strains that belong to the same phylogenetic lineage as that of these hypervirulent Mycobacterium tuberculosis. And we also determined the effect of these mutations on the neighboring genes in the virulence-associated transcriptional regulators, including PHOP gene. We found this mutation genes has an effect on the expression of the PHOP gene, and it overexpressed the PHOP gene. And even though the genetic mutations specific to the hypervirulent Mycobacterium tuberculosis strains have been determined, the causative role of these genetic mutations in mycobacterial virulence were not determined. Therefore, our objective is to determine the causative role of overexpressed PHOP gene in mycobacterial virulence. If I come to the methodology we used, in this case we used animal model, which is a zebrafish mycobacterium marinum infection model. And in order to use this model, we constructed our bacteria in the way that it is appropriate for our experiment. First, we constructed the plasmid. Contains our target gene, which is the PHOP gene in this case. And we transferred the plasmid into the competent cells of Mycobacterium marinum. And after we confirmed the presence of the plasmid inside the bacteria and the overexpression of our gene inside the transformant Mycobacterium marinum, we prepared our bacteria and then we injected it into zebrafish. Then we assessed the effect of the infection inside the zebrafish using histopathological analysis, immunohistochemistry, culture method, and monitoring the granuloma-like signals inside the body of the fish. And finally, we analyzed the data and came up with some differences between the effects of these two strains, which includes the transformant Mycobacterium marinum and the wild-type strains. The first one is differential expression of the PHOP gene in the transformant Mycobacterium marinum. And as we see from the figure here, the PHOP gene inside the transformant Mycobacterium strain is more expressed than that of inside the wild-type strain. And after we confirmed the overexpression of this gene inside our transformant, we prepared the bacterial suspension and injected it into the fish, and we assessed the survival rate of the fish infected with both two strains. And the red line here on the figure represents the survival rate of those fish infected with the transformant strains, and the other blue line represents the survival rate of those fish infected with the wild type strain, and the green line represents those control fish. And we can see from the graph those fish infected with the transformant strains have a low survival rate than that of the wild type strains. And the other difference we observed between the effect of these two strains was the infectivity of the strains. Here from the figure, from those fish infected with wild type strain, only 7% of them developed infection inside their brain. And in marked contrast, from 43 fish infected with the transformant Mycobacterial marinum strains, about 35% of them developed infection inside their brain. And the other difference we observed was the bacterial load inside the brain tissue of the infected fish. And as we observe here, we can see a more number of bacterial load over here in figure A when we compare it with the one inside figure B, which is from the brain tissue of those fish infected with the wild type strains. And the other difference was the pathological feature of these two strains inside the brain tissue of the respective infected fish. Here, in the brain tissue of those fish infected with the transformant strain, we observed these pathological changes, a kind of granuloma-like lesions. And in the control fish and those fish infected with the wild type, we could not find such kinds of pathological changes, and this is to show the presence of the bacilli inside the brain tissue where the granuloma lesions occurred. And the other most important difference we observed was the granuloma-like signals inside the transparent alive fish, and this is to show the granuloma signals inside the fish infected with the wild type, and the second row represents those granuloma signals observed inside the transformant inside those fish infected with transformant strains. And we can see the number the difference in the number of the granuloma-like signals inside the body, and at the same time we can see the difference in the location of the granuloma-like signals. In case of those fish infected with transformant strains, we can see the signal inside the brain region, but in case of those fish infected with wild type, it only approaches to the head region. Based on the results we obtained, we can say differentially expressed PHOP gene can enhance the virulence of Mycobacterium in causing tuberculous meningitis. And it is believed that this study is the first to investigate the causative role of genetic markers specific to the hypervirulent Mycobacterium tuberculosis strains, especially using animal model. Therefore, our finding may provide a knowledge basis for the development of new and effective vaccine against tuberculosis. And these are the references I used, and I would like to thank the Department of Health Technology and Informatics, the Hong Kong Polytechnic University, the Hong Kong PhD Fellowship Scheme for financial support. Thank you all for your attention. Thank you.